Good morning. I want to thank y'all for coming this morning. Princeton, can y'all still hear us okay?
Yes, Carrie, we can hear you.
Wonderful. Thanks, John. I just want to thank everyone for coming this morning. Ethan Snyder's been my master's student for the past 2 years. He grew up in Meade County here in Kentucky, and he got his bachelor's degree in agronomy from Eastern State University. And he joined my program back in, I think, May or June of 2015. He initially started on a totally different project, and we decided that it was doomed for failure after we had 2 flood events that killed all of our sensors within 2 weeks. And the producer told us they were 1 in 100-year events, both of them. And so we decided that he was no longer going to work in Muhlenberg County and that we would find him another project. And so I think it actually was a blessing in disguise, because today Ethan's going to talk to us about developing growing degree days for predicting soft red winter wheat in Kentucky. And I think this is actually going to be really important, not only for Kentucky, but also the nation. So Ethan?
Well, thank you, Carrie, for that introduction. Start off with a bang there. I appreciate everybody coming this morning, and sorry for the little bit of short notice about my exit seminar this morning. But as Carrie said, I'm going to be talking to you today about my master's project concerning soft red winter wheat growth and development here in Kentucky. And I'm going to go over first some of the reasons why the study was initiated, as well as the overall goal of the study and the objectives that we aim to complete in order to reach that goal as well. We'll go over the experimental design, materials and methods, and as well as the results and discussion from my greenhouse, lab, and field assays. And just a little background on the reason the project was initiated is that Carrie actually realized a need for a rule of thumb for estimating the number of growing degree days, or in a sense estimating the timing that was required by soft red winter wheat to reach certain developmental growth stages based on some problems some producers had. She realized they needed to have a way in which to look at some problems in the field. Producers are also often applying, or at least that they were and somewhat still are, making applications based on a calendar date, which is how the old wheat growth and development was based. It was just like, well, at a certain time of year, we're going to go out and put our first application of spring nitrogen on because it's February 15th. That's just what you do, as long as the ground's in good shape. So they, that's how they operated, and we aim to give them a little more information. Predicting winter wheat growth and development is difficult, and that's the reason that that nobody had established a growing degree day estimate for these critical growth stages in winter wheat thus far. It's complicated by the effects of photoperiod, which is the day length, and vernalization, which is of course the chilling requirement that wheat has to endure in order to initiate reproductive growth. Climatic variability might also further complicate our winter wheat management strategies in Kentucky and as well as other places too, through a couple different mechanisms. Freeze damage potential can be more severe. Well, you can have a higher chance of freeze damage in years where winter wheat's at a advanced growth stage because of a warm spring or just an overall mild winter. So if you have you'd have an increased chance of freeze damage in some winters. Less intense winters, warmer overall. Warm springs also might reduce our vernalization period, and through that vernalization period being reduced, that would limit us to to a smaller number of cultivars adapted to our state. Also, excessive spring heat is known to reduce the effective grain filling period in winter wheat. And that would obviously have a direct impact on winter wheat yield, decreasing that grain filling period. So the overall goal here was to increase the effectiveness of winter wheat management here for our producers in the state. And we aim to carry that out through the completion of these objectives. And that was to characterize 50 different soft red winter wheat cultivars and experimental breeding lines in we aim to characterize them for their vernalization requirement and photoperiod sensitivity in 3 assays: a field assay, a greenhouse assay, and a lab assay. And we wanted to develop that estimate of growing degree days for winter wheat to reach 8 specific management-related growth stages for our state. And we hope that that would also contribute to some estimates for the impact of potential impacts of climate variability on winter wheat in our state as well. And with that, I'll go right into my greenhouse assay that we completed for 2 years. We put that greenhouse assay out first at Princeton in 2016, and then we completed it also at Lexington in 2017. We planted the 50 soft red winter wheat cultivars that I mentioned into plastic pots in the greenhouse, as you can see here, and we replicated those cultivars 3 times. We planted 2 seeds per line, and grew them out to the Feeks 2 growth stage, which is about the growth stage you see here, about 2 or 3 leaves. And at that time, at the Feeks 2 growth stage, we moved the wheat out to the field for a vernalization treatment. Now you might ask, well, why wouldn't you have done that in a controlled environment like a cold chamber? But Carrie and Dave tried that a couple years or so before I started, and it didn't work out in the cold chamber, so we decided to use the field setting instead. We vernalized those seedlings outdoors for periods of 3, 6, or 9 weeks. And at that time, 3, 6, or 9 weeks, we brought them back into the greenhouse, and we measured the occurrence of Feeks 1053, which is the full flowering growth stage for wheat. We measured through that, when we measured the occurrence for full flower, we were able to calculate the number of days and growing degree days, cumulative growing degree days, once the wheat entered back into the greenhouse. And for all purposes in my study, the GDD calculation will be average temperature in degrees Fahrenheit minus 32. So as you can see here, we had some rougher looking wheat plants, especially at the later stages of growth. This was later on, about actually probably late May. So you can see, though, what I want to draw your attention to is the 3 different types of wheat we've got going on here. We've got wheat that's heading. We've got some wheat here that looks to be still growing in the vegetative growth phase, and then we've got some like this that it's either dead or very near dead. And we had a problem identifying or how a problem with how we were going to identify differences in dead plants versus those that didn't vernalize and stayed in a vegetative growth phase. So what we did was implement or rate those plants that never reached a certain growth, or that never reached 1053 in the greenhouse, as 500 calendar days and 2 million growing degree days. Now that sounds a little extreme, but it was a way in which we could differentiate between plants that never actually reached 1053 from those that were dead. So with that, I'll go right into my greenhouse data. And you can see here that I've got a table of the number of days required by winter wheat to reach the 1053 growth stage after 3, 6, and 9 week vernalization treatments. And I want to make a note that our average total length of our greenhouse trial was only 179 days. But you'll see some numbers on here that are much more than 179, and that's due to the fact to us putting that 500 in place of a plant that never reached 1053. That's how we were able to differentiate between those types of data. So first, I'll draw your attention to the significant cultivar effect on the number of days to 10.5.3 in the 3-week burn treatment, as well as the high range in number of days and the large mean number of days. Now, after 6 and 9 weeks of vernalization, you can see that mean number goes down. The range increases for 6 weeks, but we'll discuss that in just a second. But it really tightens up for 9 weeks. And I want to go over this 499 figure and the reason that we have a 499 maximum number of days after 6 weeks of vernalization. We had a cultivar, and specifically it was a Truman cultivar that 5 out of 6 plants for that variety died, and then the remaining never reached 10.5.3, so we had to rate it as a 500, and that kind of inflates our maximum as well as our range for the 6-week vernalization treatment. However, if you look at the non-significant p-value for 6 and 9 weeks of vernalization, that indicates to us that 6 and 9 weeks is in fact enough time to vernalize, and 3 weeks will show us our real differences in cultivars' vernalization requirements. And I'll draw your attention back to that 153-day range after 9 weeks of vernalization, as it's much less than 240. With that, I'll talk a little bit about the growing degree day, the number of growing degree days required to reach these 10.5.3 in the greenhouse as well. You'll see that the trends are kind of the same. You've got a significant cultivar effect after 3 weeks of vernalization, large ranges in the number of growing degree days after 3 weeks of vernalization, and those numbers tighten up as you move into the 6 and 9 weeks of vernalization, especially the means for number of growing degree days after 6 weeks and 9 weeks of vernalization. You also notice a non-significant p-value for the effect of cultivar on time to 10.5.3 and growing degree days. Now, but I want to draw your attention back to the 3-week treatment and days for our actual vernalization requirement classifications. Now, I mentioned the significant cultivar effect and the wide range and number of days. Therefore, we felt that was the best place for us to get separation between cultivars and their vernalization requirements. 6 weeks and 9 weeks is pretty much known to be enough time for winter wheat to vernalize. So in that 3-week vernalization treatment, we should see our ability is there to separate those out into categories. And that's what we've done here. This table shows the relative vernalization requirements as low, medium, and high, and the number of lines in each, and the days that were required to full flower after 3 weeks in the greenhouse. And the cultivars that were classified as low relative vernalization requirement took less than 145 days to full flower. Those that took more than 255 days were referred to as high relative vernalization requirement cultivars. And this is the list of how the cultivars shake out by, these are just in alphabetical order for each of the 3 groupings. But I want to draw your attention really to just one. Pioneer 25R32 means a lot to us in this specific instance because we know Pioneer 25R32 to require a long vernalization period. Now, since it shakes out in the high RVR classification after 3 weeks of vernalization, that kind of solidifies our methodology for classifying relative vernalization requirement. I'll also want to characterize the cultivars by their photoperiod sensitivities. So we'll look back at the 9-week burn treatment for that data. And the reason behind it is that the reason behind us using 9-week vernalization data for the photoperiod sensitivity classifications is that this is where we should see the least confounding effect from relative vernalization requirement, because you've fulfilled that requirement after 9 weeks of vernalization. and it should be, you know, more photoperiod effect on wheat at this point. So we classified our cultivars into photoperiod sensitive and insensitive groupings. We had 25 lines in each, and those that required more than 124 days to reach full flower were considered sensitive, and those that required less than or equal to 123 days were photoperiod insensitive. I'll draw your attention again to 3 cultivars in this case. Truman we know to be photoperiod sensitive, and it in fact does group into our photoperiod sensitive classification, while Pembroke 2014 and Pembroke 2016 group into our insensitive classification. We also know those to be photoperiod insensitive. So again, you know, solidifying our methodology for our photoperiod sensitivity classifications. So in the greenhouse, to conclude what we found, our characterization of cultivars into RVR and PS categories seem to work well, because we see those cultivars group into categories that exhibit similar characteristics to what we know to be true for them. Our classifications seem like they'll be sufficient for us to use in our explanation of some of our field data as well later on. We also, as I mentioned, wanted to characterize these cultivars based upon a lab assay, and that was done with a competitive allele-specific PCR. We characterized the same 50 winter wheat lines as well as a Norden hard red spring wheat line for a positive control to kind of separate from our winter lines. CASP is now a common technology used in marker-assisted selection for wheat breeding, and I want to let you know that we characterize these cultivars at PPD A, B, and D loci, as well as Vern A, B, and D, and reduced height B and D loci as well. Now I want you to bear with me as I explain how this genotypic data is analyzed normally. Results from CASP data are usually analyzed in a scatter plot just like this. The scatter plot, the values within the scatter plot, or each of the triangles, circles, or squares, represent one of the 51 cultivars that we characterized. The actual value is a fluorescence value returned from a machine that we use to run our PCR. And the fluorescence intensities, or the high, whether they're high or low, indicate presence of two different sets of dyes that return different fluorescences. Those are called FAM and VIC, and that is actually what my X and Y axis are labeled as, although I know I'm not even supposed to say this, but it's hard to see. So, and when you look at those different groupings, the different fluorescence values are associated with particular DNA sequences, and that's how you identify differences in cultivars. So I'll show you first our water controls. These are the closed circles, more towards the left side of your screen. Those are just negative water controls in order to show some separation between cultivars or between wells in the 96 well plate that had DNA in them. But I really would draw your attention also to the closed triangles and the closed squares. You'll notice that these clusters are somewhat defined, and this is what you would actually consider good separation for differences in cultivars based upon their genotype. But there's some inconsistencies in this data for what we know to be true. A high FAM value in this case along the x-axis indicates photoperiod insensitivity, and that would be the group of upright triangles there. Now we know Truman to be photoperiod sensitive, so Truman does group with the photoperiod sensitive cultivars here. However, our photoperiod insensitive cultivars, Pembroke 14 and 16, also group in that in the group of photoperiod sensitive cultivars. We know that not to be true, so we can't reliably use this data for our, for genotypic characterization. And I'll give you one more example. This particular marker is for detecting PPD B1A and B1B photoperiod alleles. Again, it's similar to the last marker, but what I want to show you is that it groups up. It looks to group nicely. You can see the group of water controls, and then you can see the group of upright triangles, which again indicates photoperiod sensitivity. But it's not reliable, since Pembroke 14 and 16 group in that photoperiod sensitive group again. And we just, after 2 attempts at the genetic characterization, there were still there's too many inconsistencies in the data to report it as a clear genotypic characterization. But even though this is pretty frustrating for me, having to learn another technology like this and get some lab experience, it was a good experience, but it was frustrating in that it would, our results weren't clearly, didn't turn out as nice as we would like them to. But that's really, one of the reasons that wheat breeders continue with phenotypic characterization as well. So we're going to continue some of that genotypic work, and there's some stuff going on right now for working on my manuscript. I'll get into my field design, or my field study, which is really the meat of my study work, where I spent most of my last 2.5 years. We aim to, we initiated the field study, in order to see how the cultivars acted in a real field environment, exposed to several different environmental conditions through different planting dates. So we had 3 site years, 2 at Princeton in 2016 and 17, and 1 at Lexington here in 2017. We planted the same 50 soft red winter wheat cultivars and experimental lines into hill plots, and we replicated those 50 cultivars 3 times. We planted 10 seeds each, as you can see me kneeling down and planting here by the flags, into each hill plot, and with the 3 replications and 3 or 6 different treatments, with the 6 different treatments being 6 different planting dates successively from October to March, that brings us to a total of 2,700 hill plots that we had to look over in 3 site years. So I'm sure, I know Dr. Pierce mentioned it, but I'm sure if you drove out Newtown Pike or drove back towards town on Newtown Pike anytime before about June 15th, you saw a lot of flags out there in one spot, in a low spot on the Spindletop farm. If you drove into Princeton on Highway 91 in the past 2 years, you've also seen a lot of flags out there, and that was actually my flag study, what some people termed it as. But these 2,700 hill plots were really the only, that was the way that we were able to measure time in growing degree days and calendar days to these specific growth stages that we were interested in. And we were out there 3 to 4 times a week in the spring and early summer months, and even up to a time or 2 a week in the winter months, checking on these, for the occurrence of growth stages in this weed, as long as the temperature was above freezing. I'll talk about the 8 growth stages that we captured or observed here briefly. Emergence, of course, you want emergence for a baseline of when your plant starts in the vegetative phase of growth. We took occurrence of Feeks 3, which is associated with a nitrogen application here in Kentucky for winter wheat, and it's also the time at which the wheat fully tillers, or it produces shoots off of the main stem that are also capable of producing heads for increase in grain yield. Also, we measured Feeks 5, which is pseudo-stem erection, when the plant starts to stand up, upright. But at that point, the growing point is still below ground. It's still a vegetative stage. Nitrogen is also usually applied if you do a 2-shot application, you usually apply nitrogen your second time at Feeks 5. Feeks 6, however, is the first reproductive growth stage that we measured occurrence of, and it's when the growing point comes above the ground. The weed head actually comes above the soil surface, and it's highly susceptible to freeze damage from that point forward. We measured occurrence of Feek's 9, which is flag leaf. And that's the flag leaf is important because it really provides photosynthate and photosynthesizes for pushing assimilates and things to the grain head during grain filling. We also measured the occurrence of Feek's 10, 5, 1, and 3, which are beginning and full flower. That's and the anthesis period, and those are also important for fungicide applications for control of Fusarium head blight. That's often when we do that here in Kentucky. We also measured, of course, the occurrence of harvest maturity, which is Feek's 11.4. And I want to bring back up real quick that 500 and 2 million growing degree day ratings, because we see such stark differences here between our different growing or different planting dates for their growth stage at a particular point in time. You'll see this picture in late May has wheat that was planted in October, wheat that was planted in January, and then the red flags are wheat that was planted in March. And the March planted wheat at the end of May here is not even exhibiting reproductive growth. So we had to, again, determine how in some of the cultivars or some of the planting dates where it wasn't so clear cut, where there were only a few varieties that produced heads, we needed to be able to tell the difference between those that were not able to reach or a certain growth stage and those that were dead. So I'll show you some data from across all 6 of my planting dates here, and this is number of growing degree days calculated to reach for cultivars used in this study to reach the certain 8 growth stages that we measured. I'll draw your attention first to the significant cultivar effect on the number of growing degree days in every growth stage besides emergence of Feek 6. And then the highly significant planting date effect, which we would totally expect with 6 different planting dates that range from October to March. Also want to mention the high range. This is much like the greenhouse data where we've got those 2 millions input. It influences a very high range in number of growing degree days required by wheat to reach those reproductive growth stages, in particular where the 2 million starts here at Feek 6. And although as an agronomist, although as this is the data across all 6 dates is very interesting, and it's good for use in some of these climate variability impacts that we're interested in looking at, as well as breeding efforts and maybe some modeling in the future. I'm an agronomist, and I would be much more interested myself in October and November planting dates, as that's what's more normal for Kentucky. I will say that our November planting date was a little on the late side for what's normal to Kentucky producers, because it was more like the 3rd week of November. That was just due to the fact that we wanted to space those plantings out, though, by month. So just keep that in mind. It is a little late. I'll draw your attention again to the highly significant planting date effect on the number of growing degree days to all growth stages measured, and the significant cultivar effect as well, which we still expect to see some cultivar effects on number of growing degree days based on some relative maturities. But you really see that this range has actually, really decreased. The range in number of days between October and November wheat planting dates is not, it's not nearly as high. It's tightened up a lot. Since we characterized I mentioned that in the field we wanted to also use some of that greenhouse data that we were able to come up with, the relative vernalization classifications and photoperiod sensitivity classifications. We wanted to use that to help explain some of our field data. So that's what we've done here. I've analyzed the number of growing degree days required by wheat cultivars that group into these high, medium, and low relative vernalization classifications. We analyze them separately. This, data saying that relative vernalization requirement and photoperiod sensitivity has an effect, within each of these normal planting dates for Kentucky would be, it would make a lot of difference in our development of a growing degree day estimate for wheat to these specific growth stages. So that's the reason why we wanted to check and make sure that there, overall are not a lot of differences in relative vernalization requirement and photoperiod sensitivity in a field setting. When you plant within a relatively normal planting date. So what I want to point out here is the non-significant p-value at all stages besides the non-significant p-value for the relative vernalization requirement on growing degree days to all stages besides Feeks 3 and 9 and 10.5.1. But we'll talk about 3 and 9 right now. Although there are significant differences detected, and we're looking at growing degree day differences of 8 growing degree days there. That didn't actually identify as a significant difference when we ran Tukey's mean comparison or Tukey's adjustment for mean comparison on this data. So overall, it seems that those differences don't really matter. When you look at the 10.5.1, the significant effect of relative vernalization requirement on number of growing degree days to 10.5.1, though, it's pretty significant, and we actually need to look into that just a little more to make sure that that's what's going on there, whether that is a relative maturity difference or not, and we plan to do that for my manuscript that we're going to write up. So I explained to you how we analyze field data based on relative vernalization requirement. And you can see here we've done the same thing for photoperiod sensitivity classifications. And again, there's a significant effect of all planting date on all growth stages, even in October and November. But there is not any significant effects of photoperiod sensitivity or the planting date by photoperiod sensitivity interaction on the number of growing degree days required in these two different photoperiod sensitive classifications. So really what I'm trying to say is that overall relative vernalization of these cultivars, whether high, medium, or low, or photoperiod sensitivity, whether sensitive or insensitive, doesn't seem to matter in these October and November planting dates. And therefore it helps us support our goal of creating this growing degree day scale. And that's what I'll go over with you next. And although Kerry was actually wanting, when the project started, to have one growing degree day estimate for across both October and November dates, since our dates were spread out over a month, you're going to see differences in growing degree days when you have 30 days to accumulate heat units. So we had to separate these out into two growing degree day estimates for one for timely planting date and one for a later planting date. That would be a fairly late planting date. And I'm going to get into some ground truthing for these estimates here in just a second. We gathered historical data for ground truthing from both the Kentucky Grain Crops blog and from some of Bill Bruning's wheat variety trials data as well. And we gathered data from seasons we knew to be pretty much on schedule for winter wheat growth, delayed seasons such as 2014 when we got later on into the spring and wheat wasn't even starting to show signs of standing up and getting into that Feeks 5 stage, as well as an advanced growing season like we had this past, where wheat was at Feeks 6 in March and highly susceptible to some of those freezes we had. We also used some of Bill's data from 2008 to 2017 in order to get some mean heading dates and harvest dates for cultivars that we specifically used in our study. We used only data from cultivars that were used here. And this is data actually from the Grain Crops blogs and the Wheat Science newsletters. And you'll see the growth stage that was reported, as well as the date of occurrence for that growth stage, and then the growing degree days that it took wheat. The growing degree days that we calculated roughly that it would require to reach those growth stages based on historical weather data from that year. And we've compared this to our estimate of growing degree days here. You can see for Fix 3, there's about a 260 growing degree day difference. But as you move into this normal year in 2016, where wheat was at the Fix 6 growth stage, it's a little more accurate representation of how many growing degree days it takes wheat to reach this stage. And in the Fix 6 growth stage for March 1st of 2017, we're only off by about 100 growing degree days on our estimate of how long it took wheat to reach that growth stage. So it seems fairly accurate in these early growth stages. But here we'll look at some of the later growth stages. We've got data here on the left from Bill studies, which are these are the mean number of growing degree days that we calculated, of course, with the October 15th planting date. The mean number of growing degree days required to harvest. This is all based on some historical weather data and dates of occurrence for harvest and heading. We compare that to our growing degree day estimates. You'll see here that I'm, we've got beginning flower and heading. Well, we didn't report heading in our study, since heading is not necessarily a management related growth stage in Kentucky. Very important, but not management related. So what I'm going to do is say that in a 3 to 4 day period that normally occurs between heading and beginning flower, you're going to accumulate about 130 growing degree days. So if you add 130 to Bill's data, or to the growing degree days calculated here in Bill's variety trial data, we get fairly close for our beginning flower estimation of growing degree days. And the same goes for harvest. We're within 230 growing degree days there as well. So when we developed this estimate, that was my goal. But we were also able to collect some useful data from those growing degree day, from the other planting dates for estimation of some climatic variability effects on winter wheat production here in the state as well. Most studies currently examining climate variability on wheat are really concerned with anthesis temperatures and grain filling temperatures and their effect on wheat yield. So I examined the average daily temperature from the anthesis period for each of the 6 dates that we put out, and I've reported these here. But really, I want to show you that as you go forward in time from the October to March planting dates, you have an increase in average anthesis temperature in all but one case. But you see here that anthesis only lasted for 2 days in March of 16. That was due to the fact that we only had a couple cultivars out there that actually flowered, so it really tightened our variability up. But I want to show you now some regressions that we did on the on some of the relative yield components that we measured out of the field. And we measured total seed count per plot, total seed weight per plot, total number of seed heads per plot, and 1,000 kernel weight per plot, and did that on a relative basis compared to that of what we measured from the October planting date. Now, when you move forward in time from October to November, December, and so on, you see the temperature increases I've reported here, and these are the anthesis temperature increases. So as we increase with planting date, or we delay planting date past October, this is showing us that that we have a pretty strong linear trend, negative linear trend in relative seed count with each successive planting date. We also have the regression done for relative seed weight as well, and that's very interesting as it seems to, we've got a high R-square value and a highly significant p-value as well here, and it shows the same trend, negative effect on yield as you move to the later planting date, or not yield, I'm sorry, but this relative yield component that we measured here. And as you'll notice, about each 1 degree Celsius increase on our scale from October to November, average temperature in Celsius increased by about a degree. You'll see somewhere between a 10 and 20% decrease in yield. Now that's pretty interesting. You see the same thing for relative thousand kernel weight, with a very high R-square value in this case, and a significant p-value for this regression. And as you move forward in time, you get lower thousand kernel weights with delayed planting dates. Same thing with relative head count, although we see a little variability here, and it causes our R-square value not to be quite as high, but a little variability at Lexington in 2017 due to some of those early spring freezes we had, some environmental effects there. But what I really want to say is that overall, the delayed planting date effect on the relative yield components we analyzed is strongly negative as we move forward in time from October to March, which we would expect, with a delayed planting date. But we also want to relate that to these antithesis temperatures. It sure seems to be that that as you move forward in time from October to on, that you have decreases in all these relative yield components for with that. Our data indicates about between a 10 and 20% decrease in relative yield components based upon each 1 degree increase. But the FAO actually they report a 1 degree Celsius increase in temperature during grain filling will decrease yield by 8 to 10%. So we're seeing some stronger effects here, but keep in mind this is not yield on a field size scale. This is just data that we're collecting from hill plots. But it is indicative that we're going to have some we can have some major changes. So in conclusion, remember we initiated this week study to identify the growing degree days required to specific wheat growth stages. And in that, we were provided a cool opportunity to work in some other disciplines, including the genotyping work. And although it didn't pan out, that's just that's how it's been so far. But we also were able to get some good information for increasing our knowledge on climate change through the data collected in the later planting dates that I reported with the yield component differences. And we think that we have made we can make a relative contribution once we publish this data on what we see in those relative yield component decreases. The greenhouse assay goal, was of course successful with characterizing the relative vernalization requirement and photoperiod sensitivities of my cultivars. The lab assay goal, as I mentioned, was not so successful, but we're hopeful for what's going on now with genotypic work. The field assay goal, of course, we successfully determined that we needed 2 different growing degree day estimates, and this definitely supports our goal of helping Kentucky producers, and hopefully moves us towards another goal of associating specific management decisions with a range of growing degree days. And it certainly seems to be applicable here in our state. We've also had a little bit of national interest in the scale as well, for use of such a system, since nothing exists elsewhere. Well, I mean, there's some growing degree day estimates in some other places, but they're spring wheat related. So with that, I'd like to thank you all for your time today. I'd like to thank my committee and all my lab mates that have helped me along the way, as well as our technician who's retired now, John, and all the undergraduate students and other graduate students that I didn't list here, that have helped me in many ways, from inspiring me to keep moving when I didn't want to continue, or study with me on a test, or influence me to go work out, whatever it was. Thank you all very much. And with that, I'll take a few questions if you have any. Yes, Anthony.
All right, so going back to your CAS assays.
Yes, sir.
You mentioned that you looked at several different loci for both Photoperiod and Vernalization.
Yes, sir.
And seem to be disappointed, or kind of like downplaying the fact that by looking at any one of them, you weren't able to predict the kind of like the field sensitivity or insensitivity for Photoperiod, for instance. But since you know that there are several different loci that control that trait, would you expect to be able to predict it by just looking at them one at a time?
Since we know there are several different loci that affect that trait, will we expect to be able to see differences in one assay at a time? Well, our impression was that we should have, or my impression was that we should have been able to see, like Pembroke 14 and 16 shake out as what we know to be true about them in a particular assay, but maybe.
But what I'm asking is, were those Pembroke ones, were they sensitive or insensitive at all, the loci that were?
Yeah, we actually, and the reason I chose to just to press it down to only two of these markers was that we even looked at the PPD-D1 loci, in which Dave indicated was very a more robust example of how, or a better marker, a better loci, or better representative of indicating the genotype, and it showed wrong as well. I mean, it showed Truman in I believe it showed Pembroke. I know it showed Truman in insensitive, as characterized as insensitive. So we couldn't make out that it was successful. Myself, maybe we need to revisit once again. Does that answer your question?
Sort of. I mean, I guess I'm just making the point that it may not have been it may be just more complicated. That if you have several different loci that are controlling a trait, maybe it simply needs to be, it can be the insensitivity, or could be caused by any one of them. And so with the others, it could be sensitive allele. It wouldn't matter because the insensitivity is being caused to the different loci.
I understand what you're saying. So we need to make sure that each cultivar that we're making an example of is in fact sensitive or insensitive at each of.
Yeah.
I understand. Okay. Well, we need to look into that further for sure. Thank you. Yes, JD.
So if I understand your conclusions here, you feel pretty comfortable that based upon 3 site years, the growing degree days assessment is enough to feel pretty confident to make that recommendation to producers to use that for making these decisions? Or do you think there's some additional environments need to be evaluated to further refine those calculations?
Personally, I think we've got a good start. I think, as in any scientific data, especially that is influenced by environment that we can't control, I think we would be it would be a very good idea to have more stock years. I don't think, obviously it's not as fine tuned as what we've got for corn. Corn is pretty fine tuned to, of course they also, breeders release it as, as this cultivar or this hybrid takes X amount of growing degree days to reach a maturity. So I think if it was evaluated at, it would definitely be a good idea to evaluate it more in different environments, and that it, for sure needs some more fine tuning before we get as specific as such as what corn is. Yes, Mitchell.
To add to what Dr. Green was saying, based on your data and what you found in the field, and the massive amount of data and all the conclusions you're trying to make, do you think that there's a reason why farmers are going off the calendar date? Does your data say, okay, well, that's probably over time why they chose this date to make a targeted application of the N or spray date?
Well, I mean, they made those, they've definitely made their conclusions over time by seeing what happens in the field every year. And there's probably a lot of cases where they're pretty warranted to make that application based on the time of year that they think it needs to be done. But in certain, I think what we want to get out of this is in certain odd years, when calendar date doesn't apply, when calendar date is off by 2 or 3 weeks, which it was in some cases, like this year we were off and we were ahead of schedule by at least a couple weeks, and then in 2014 we were behind by a couple weeks, and it still seems to apply, in that situation. Like you're still at that growth stage, but you're at a different calendar date. So just a better, a more informed decision. Hopefully they'll be receptive of it, and that it's just another way to help them make decisions.
I want to see if there's a question at Princeton.
Does Princeton have any questions? I think they cut that off actually because of winds.
Yeah, they did.